Okay, well, welcome everyone to our weekly DMICE Research Conference, although this week we're actually having two conferences, so this one is today on Tuesday, and then also we'll have our regularly scheduled one on Thursday. Our speaker today is Janice Patterson, who is in the bioinformatics and computational biology track and is going to be defending her master's thesis proposal. So for those who aren't familiar with this process, she will give a presentation to all of us, and then there'll be some time for questions. The public will then be dismissed, leaving behind her committee, who will ask her additional questions, and then she will be dismissed, and the committee will determine whether she's ready to go on and do her master's thesis work. Janice comes to OHSU by way of PSU, where she has a bachelor's degree in biochemistry, and she's currently working in the lab of Dr. Michael Heinrich, who's in the Division of Hematology Oncology and also at the Portland VA Research Center.
So my proposal today is called Understanding What's Under the Hood, Increasing Accessibility in Omics Results. So I'll begin by starting to talk about my motivation and background, as well as my aims, and which will include two use cases, and some preliminary metrics and the impact and next steps of my project. So my project really stems from this problem of big data. This figure emphasizes the problems with combining patient data from clinical information to patient background history, and really how diverse and complicated the information is, and how you need increasing ways of processing and transforming, like increasingly complicated ways of processing and transforming the data to make meaning out of it and make sense out of it. And really it's about, and ultimately when you get your results from it, it's about blind trust. And really you don't understand, like, you don't really know whether the transformation of the data kept the fidelity of the data. And this figure doesn't even include the omics part of it. So so a lot of the genomics and like the proteomics part of patient information. So this quote by Tukey and Wilk, I think, kind of prophetically emphasizes one of the big problems with big data, and he emphasizes the importance of exposure of your data and how difficult it is to expose data for human analysis. And this problem of exposure is really a big problem with like large data sets, because you have these files that you can't even open with like a word processor or text processor, so it's really hard to get your hands around the data and really expose that data. So one of the large problems that has come with this problem of big data is this problem of reproducibility, and this figure shows is a Bayer study. And it shows, and they attempted to reproduce 67 in-house published data. And they found that only 25% were reproducible, and while 65% had inconsistencies between the published finding and the reproduced finding. So this problem of reproducibility increases with the increasing amount of data that you have. So it's easier to make mistaken assumptions about the data when you can't expose it, as well as the multiple transformations that have to happen to the data to make anything meaningful or interpretable from your data. So how do we get underneath all this data, or underneath the hood, so to speak? So, and one way is to change how we interact with it and how we really think about it. This figure representing the gulf of valuation and execution kind of represents this gulf, these gaps that we have between what we conceptually think as well as this world of data. And the gulf of evaluation represents sort of the gap of interpretation, the effort that we have to do, that we have to put in to interpret the data and whether that data has met our expectations. And the gulf of execution kind of represents the gap of usability and between what we want and what the data in the world can tell us and what we can do with it. And you can see that even for my toy example, that there were two transformations that had to happen to the data to get something meaningful, interpretable, that answered our question. So increasing the interaction of data can really narrow this gap. And not only do you narrow the gap so you can directly see the impact of these transformations, and you immediately get feedback of these transformations by exposing the data immediately with interactive data, like visualizations and things like that. And it makes data more accessible and interpretable ultimately. So interactive data has extensions beyond static data. And this table classifies the components of interactive data. And obviously, you have being able to view and visualize the data. So that's one of the components. And then interactive data has also the ability to manipulate your data. You can filter it. You can filter it, verify assumptions, as well as diagnose your data as well. And this process and provenance also lets us. It's a way of interactive data visualization lets us view the processing of the data directly. So you can kind of see the story of what happened, of the processing that occurred to the data by looking at the visualization as well. So you can do, as well as sharing your visualization with others, and they can see the processes that you implemented and the transformations that you implemented on the data. So my aim one will be to develop these metrics and visualizations and statistical summaries to assess the robustness of computational methods and algorithms in high throughput sequencing. And I'll really be focusing on downstream analysis, computational methods and algorithms. And my second aim is to implement these in two use cases: copy number variation and chromatin immunoprecipitation. So my first aim is to develop these metrics to assess the algorithm and computational reproducibility. And these are really general metrics. So they're for high throughput sequencing. So that assess reproducibility and basically the validity of your method. And I'll do this in an interactive context. And so basically thinking of ways to basically use interactive visualizations to implement these, as well as integrate them together with other metrics. So what is high throughput sequencing or massively parallel sequencing? I made this figure just so you can get a feeling of what the results look like. And so you take DNA, and in this example, you take DNA and you fragment it, and you put it into the massively parallel sequencer. And what you get from it are millions of 50 to 100 base pair reads. And these are aligned to the reference genome, usually by species. And you can see how fidelity and how crucial this step is really to any downstream processing, anything you're going to be trying to find afterwards. This is a crucial step. And you'll get metrics like read depth and read coverage. And the above example, I have a pointer, read depth, kind of shows an example of read depth without coverage, so or with minimal coverage. So you have 6 reads basically supporting the alignment at this small region of the genome. Well, read coverage is kind of an example of a read coverage without read depth. It's the breadth of the nucleotides that are covered. So you have all those. You have the whole sequence, the whole region area being covered, but you only have 1 or 2 reads to kind of support it. So once you've aligned to the genome, depending on the type of sequencing you're doing, you end up doing this computational analysis. And that gives you this list of results with some level of significance or confidence level at the minimum. And there's multiple computational methods that for every type of sequencing, RNA-seq, DNA-seq, and all of them use different algorithms, statistical methods, and that has resulted in a severe discrepancy in your results. So you'll have discrepancies in different results based on your computational method alone. So why do these analyses differ? It's because, like I said, algorithms differ, and the assumptions of those algorithms differ. Statistical models, whether they assume the normal distribution or things like that, correction methods, and arbitrary thresholds for statistical models, as well as parameters, whether they or if they even include a background, or how they include that background or baseline or control. Moreover, there's quality metrics at each one of these steps. So if the reads from your alignment you have FastQC. They tell you the quality of your alignment, the reads from your sequencer. And there's a whole suite of tools that tell you your percent aligned or you're uniquely mapped, but there's really nothing to tell you the quality. There's no exploratory data analysis for the results, and really you're kind of guessing at the robustness of your computational analysis. And this is my project. So my framework will attempt to assess, but my framework is basically to do exploratory analysis, find metrics and visualizations that will test the robustness of the computational methods from the results. And I'll do this in an interactive context. So using visualizations and metrics that you can interact with and really get your hands around the data. And I'll do this obviously, and like I said, I'll do this using the interactive dynamics, being able to view and visualize, manipulate your data, diagnose your results, as well as the idea of processing and provenance as well. So my second aim is to implement this framework for 2 use cases, copy number variation and ChIP-seq. And these are, at this point, I'll focus mostly on developing these use case specific metrics and visualizations, and really to assess it. And because all these types of sequences are looking for different kinds of things, and so so you need to really develop use case specific ones as well. And I can kind of integrate them with the general metrics as well. And I'll use the interactive framework Shiny in R for this, which is a web app framework. Now, I think really even the aim 1, I'll be developing these general metrics, and I think they're better understood if I explain them in terms of the use cases. And I think they'll make sense. They're better understood if I explain them in terms of my 2 use cases, which are copy number variation and ChIP-seq. So I'll start by giving you a little background on these 2, as well, and then I'll go into some general metrics as well as use case specifics for both of them as well that I've kind of explored. So copy number variation tries to find large variations in the genome associated with disease and negative phenotypes. And ChIP-seq identifies these genetic locations associated with protein. And differentiation at these genetic locations ultimately causes differentiation in the protein processes, and of course differentiation in genetic regulation and structural changes, which ultimately cause diseases. So copy number variation are these large, greater than 1,000 nucleotide regions that vary in the genome. So on the first example I have is where your reference matches a sample, and it's kind of this is what normal looks like. And then a copy number loss is shown by this deletion, where you have your reference gene, and then your sample is missing that chunk of genome. A copy number gain or duplication or insertion can either be a duplication or insertion, where the gene is either inserted or duplicated on the sample, or it's inserted into another part of the genome altogether. So there's various methods to detect these copy number detectors. What I have here is a read depth method. And so you're looking at these 50, 100 base pair reads, and you're trying to find these large ones, right? And so you're looking at read depth, and you can see that this is a copy number loss. So there's a significant less read depth in this region. As well, different copy number detectors also quantitate copy numbers differently as well. So they use different computational methods, as well as algorithms and different assumptions, as well as the statistical model for these detectors vary significantly as well. So significance thresholds, assumption and distribution assumptions. So this variation has led to some severe discrepancies in copy number detectors. This figure shows 4 copy number detectors with 33 exome samples. And you can see that they're in concordance, like the match up here, means 50% overlap of these copy number variants needed to be overlapped for concordance. And you can see there's a significant amount of discordance as well as, and you don't really know what's important. I mean, yeah, you could take this significant all the overlap of 773, but like Tuki said, that's not likely to tell you the unanticipated parts of your results. So what I have here is my mock-up of what I built in Shiny. And this is depth of coverage. Is that working? Depth of coverage up here. And the blue region, this blue bar here, is connected to this range right here. Sorry, I think I'm running out of batteries. So as you focus on the depth area, this region right here, the Venn diagram is kind of it will be reactive, and so the number of so it's basically showing the number of copy number variants at that location, at that blue bar location. Yes, I'm dead. Sorry. At that blue bar location. So does that make sense? So let me show you in an example. So as I focused in on this read depth region that has no read depth, you can see by the box plot above. There's no read depth in this region. So the number of copy number variants called by this method as this method not only decreased significantly from the prior one, the number of the overlaps of the copy numbers called also decreased, showing that this low signal region not only had less calls, it also produced less reproducible signal as well. And in this slide, I focused on a region with higher read depth and higher coverage, and you get more overlap in the number copy number variance that you call as well. So you get more reproducible signal ultimately too. So chromatin immunoprecipitation is a more targeted form of sequencing. In this little brief protocol that I have here, you have the DNA that's bound to the protein, and then you fragment it, and then you use immunoprecipitation which uses a specific antibody, excuse me, to bind a specific protein. And so the antibody is usually bound to some kind of medium, like a silica bead, and you end up trying to use the specific antibody to fill out the DNA and protein complexes that you're interested in. So you purify this complex, and then you end up isolating the DNA. And so you can see how the only reads that you should get should be from the parts of the only DNA reads that you should get should be the ones that interacted with that protein. So your alignment profile ends up looking like this. So you get this. Your alignment profile looks like a bunch of stacked reads on top of each other. And so here's your reads, and what you get are these peaks of signal. And so these algorithms or computational methods to find these for ChIP-seq are called peak collars. And you can see how read depth is a really important statistic at this step. Now, if you don't have enough read depth, you're not going to be able to call these peaks because they won't be basically stacked on top of each other. These computational methods also have a problem of overfitting as well. So if your peak detector is too sensitive, not only will you call this an obvious peak, as my husband called it, Dubai versus Detroit. So, not only will you see Dubai, this peak A, which is obviously a peak, but you'll see the 5 peaks, or however many in area B, which only have one or two reads really kind of supporting them as well, which is more likely noise than anything else. So there's variation in these computational methods as well as experimental sources of variation, like antibody, as well how specific the antibody is, as well as fragmentation, as well as what you're trying to find in the first place. So transcription factors, which is the red peaks above, CTCF is a transcription factor, are only like these 200 to 300 base binding regions, have these 200 to 300 binding regions. So you can see them as like really sharp peaks. And then another spectrum, histone modifiers have these wide binding regions, which are like kilobases, thousands of bases wide. So the variability in these ChIP-seq peak detectors has really caused the variability in the results. So on the left here, you have 12 peak collars, and this is just a count, number of peak collars that each one of these peak collars called, and for 3 transcription factors. And you can see that it's completely random, that there's the number called, there's no pattern in the number called or anything like that. And on the figure on the right, you have a single binding region for STAT6, which is also a transcription factor. And the binding motif is on the bottom here. And so this is kind of what you're supposed to see. And then above it, you have 11 peak collars as well. So not only do you get this variation in the number of peaks called, you get this complete discrepancy in the binding region identified as well. So one of the use case specific metrics that I plan on developing is this fraction of reads in peak. And so in a perfect IP, you would isolate only that specific protein, right? So it's like perfect IP. And all the sequencing reads from that ChIP-seq should be only from that peak, from the peaks itself and from signal. So the Fripp score is usually tells you like the success of your experiment. It's over the whole genome or over the whole sample. So you would expect it to be kind of high. Like for example, if you looked in this window, and your peaks and your signal was this peak, and maybe this tiny little peak here, your fraction of reads in this window is pretty high. So it's a good diagnostic on the quality of whether your immunoprecipitation worked, as well as like whether but a low Fripp score could be a failure of IP, or like maybe you didn't, or because your peak detector was completely horrible or something like that. So it's really a metric of the success of your experiment. So this is one of the general metrics that I've developed, and this irreproducibility discovery rate really addresses one of the main problems in comparing computational methods and algorithms, and it's really because they all use different statistical threshold. So a p-value, q-value, log-fold threshold, and they're kind of incomparable. And Lee et al. Suggested a solution to this using this irreproducible discovery rate. And this attempts to find a threshold where signal and it uses biological replicates, and it attempts to find a signal where replicates no longer agree with each other. So it's assuming that like reproducible signal is a true signal and all noise is not reproducible, right? So in this plot here, you have rank proportion. It's proportion, that's why it's a decimal, and an irreproducibility score. And basically, perfect concordance is represented with a straight line with a slope of 1. No concordance or correspondence between the replicates is represented with an x-squared curve. So you're trying to find this transition from where the straight line turns into a curve, or where these biological replicates go from reproducible to not reproducible. And this alternative method of finding this significance threshold for samples is more robust than like an arbitrary alpha of 0.05 or 95%. And it's because it's derived empirically from your samples itself. So this figure shows how I'm going to use IDR to explore the robustness of these algorithms and computational methods. So biological replicates will be analyzed in parallel, separately but in parallel. And then multiple algorithms can be combined, and then the number of hits basically found by each one of these algorithms can be compared as well. So if like one algorithm produces like 10 hits, while another algorithm produces 10,000, and both had an ID at their IDR, so you can really get a sense of the sensitivity of that algorithm, of both algorithms at that point, as well as other general exploratory metrics using this IDR method, that threshold versus the arbitrary threshold by the algorithm. And this interactive component provides a way of kind of optimizing your IDR. So you not only can, you can tweak your IDR basically. So you can, let's say you have a known positive, and for one of the algorithms, it was beyond that threshold of IDR, and you want to tweak it so it's included. So you can try to tweak your algorithm or your IDR and set it lower for that algorithm, and so your known positive is included in all the algorithm. It's kind of a way to tweak your sensitivity, and so you can get a fair comparison of all these algorithms and methods. So the impact from this interactive framework, it will ultimately to increase accessibility in data, in the data, as well as the methods that we use to analyze the data. And we'll use metrics and visualizations to like expose this data for analysis. And ultimately, I'm hoping it'll increase reproducibility as well as credibility by directly associating criteria that you use to filter all your data or transform your data and being able to source it and reproduce it. The framework will also provide a way of comparing computational methods and algorithms as well. You can really see how you can assess the strengths and weaknesses of all these algorithms and methods. Go. So next steps would be the possibility of integrating in with Galaxy. Ultimately, I could probably think of using outputs like get case, depth of coverage, or things like that that are known, and formatting it so you can just directly input into RShiny and get the visualization. I think that would be the easiest method. And then as well as user testing, which is essential for the usability of this. And collaborative models are important because you can see how these exploratory metrics would be essential for larger and larger data sets where you have collaborative studies. And so sometimes you'll have multiple people trying to use the same interactive visualization. So I think a robust method for that integration of multiple users is pretty important, as well as examination and other sequencing workflows, and this problem of reproducibility is more than a CMV or ChIP-seq problem. And on the figure on the left shows RNA-seq differential expression methods, and it shows 5 differential expression methods. And you can see what I think was most disconcerting about this diagram is a big fat 0 in the center. But it's RNA splicing, which is like a difficult problem to detect in the first place. So there are some concerns here, as well as mutation collars for DNA-seq. These are this paper assessed 4 mutation collars, and you can see that sorry, it's a difficult figure to see, but there's discrepancy in the number of peaks, the number of mutations called, as well as the proportion called as well. And that's it. And I'd like to thank my committee, Shannon, watching in DC, and Mike, my boss, Lucia and Kamal, and Heinrich Lab and Carbone Lab, and DMICE and friends and family. Yes.
Interesting topic. So I'm wondering about all the parameters for each of these methods that you're using. So would you use your metric to also compare different parameter settings for, say, different differential expression or whatever?
I think it's more of an extension for RNA-seq and as well as mutation colors. They're definitely applicable. So you can kind of see how depth and coverage are kind of universal. And that's part of one of my first aims, is to develop these general aims so potentially you could probably apply it to these other methods as well. Does that answer your question?
Kind of, but I mean, even for the methods that you like copy number stuff that you talked about, like every one of those methods, every one of those algorithms is going to have 5 or 10 different parameters that you could change, and then you could do a bunch of different ways of aligning it, use different aligners with different parameters on that.
Well,
so there's just a huge number of
there's a lot of variation. Absolutely. Yeah. Well, what I wanted to do is just come to the baseline. So, like, for example, there's only so much I can tweak about every single. Like for ChIP-seq alone, I think the last count in 2012 was at 35 peak collars, different kinds of peak collars, which is huge. You can't do every single parameter for every one of those, but you can kind of get them on the same basis, so you can see the benefits and pros and cons of each. Like the IDR, lot of these methods use their own statistical program or FDR or statistical method to filter. If you take that statistical aspect out and then filter by IDR, you can just test the algorithm that you use and just kind of get them on the baseline and see which one performed better there, just by taking the full spectrum of signal instead of the filtered and the queued and corrected. And so you can tweak you can get them on the same baseline, I guess, and really get the core components and try to compare them instead of trying to find all the parameters, obviously, because there's all the defaults, right? And they're like 1,000,001 defaults that are hidden in there, so it's hard to
change with different versions.
Absolutely, yes. And so you can only do so much, but I think there's, at the core, I think the computation, you can really assess robustness there and kind of go from there as well. Cool.
Do you have a gold standard by which you're assessing the quality of the agreement and the quality of the disagreement?
Well, for like copy number and stuff like that, everyone kind of uses a different method. Like I know for copy number there was 50% overlap or 50% overlap in terms of concordant exons, and required 50% overlap of that exon, and then they just counted the number of concordant exons. Does that make sense? So there's a lot of ways to measure concordance, and I think just when you're comparing them, just consistency is all you can really hope for, and as well as consistency in comparison. So if you know from your known data set.
I guess along those lines, are there any data sets for which people have actually found something and verified that they found something, and then you could use? I'm wondering if there's pre-existing data that you can use where someone has experimentally verified.
Well, you can simulate the data as well, but you can simulate data as well as with ChIP-seq. Though you have known motifs, where people have cataloged all these binding regions, and you can use those as a basis for how the accuracy of whatever your peak color defined as well. So there's measures of accuracy that you can do without actually having to simulate data or things like that.
Actually, that was the exact question that I was going to ask. So I'll extend it, and I know this may go beyond the scope of your work, but there's a lot of areas of informatics where we, a lot of testing of algorithms is based on some sort of gold standard, and we try to optimize the algorithm or use learning algorithms or whatever to achieve that optimization. So here, I assume at some point you will receive something that might be the right answer. And then how will that be used by the bench scientists to advance this work? I mean, I assume you're looking for things that a bench scientist can then study further. How would someone use the result if you could find the right answer?
Well, that's interesting. Well, there's obviously best practices in the genomics world, but you can use those best practices. I know Broad has a whole system, and Broad GATK pipelines are a gold standard a lot of the time, and it just you can use these measures of accuracy, but ultimately what I see from this framework is it's a way to assess how accurate your method was, right? You can have this gold standard, but ultimately, for different kinds of whatever you're looking for, it might not work that well. For example, using the transcription factors versus histone modifiers, those peak regions. If you use, you can see how algorithms optimize for transcription factors or these tiny little peaks wouldn't perform so well on these large peaks. So I think it ultimately a gold standard for the one use case isn't necessarily the gold standard for all use cases, and you need a method to basically assess how to assess that, though. So you don't really know until you realize, oh, why is my data, like, why do I have, I should only have 1,000 hits, but now I have 200,000 or 500,000, and it's like things like that kind of flag your data. And that really gets into the exposure, and you can't really, like, know what it did until you actually run it and see that it's inaccurate and things like that. That answered your question, right? Okay, right. Okay. Anything else?